Uh, Hello, everybody, and welcome to our first session on single-cell biology of mammalian organs. And we'll just cut straight into the talk. So first up is Ana Domingos from Oxford, who will be talking about sympathetic neuroimmune diversity and obesity.
Hello, hi. Thank you so much, the organizers, for inviting me to give a talk here at the Keystone. And my talk will not be focused on the single cell techniques, but rather how we have used single cell techniques to learn more about a system that it's not an organ, but it's a system that connects multiple organs, and that's the sympathetic nervous system, of which we see an example here. So essentially these neurons here that are labeled in red. And I'm also going to talk about neuroimmune interactions with these nerves that in the end, they will control the fate of the adipose tissue. So the tissue where we store fat and which determines when it's big, if one is obese or not. So body weight of homeostasis, it's largely controlled by a hormone leptin, which is produced by the adipose tissue in amounts that are proportional to the amount of fat. And in this way, the brain knows how much fat exists in the body. So the more fat exists, the more leptin exists in circulation. And this is the afferent arm in the neuroendocrine loop in which the brain senses leptin and therefore knows how much fat exists, which is produced by the adipose tissue, which is in fact a gland, an endocrine gland. And once there's enough fat, the brain triggers a plethora of responses that includes suppression of food intake, which in turn reduces the amount of adipose tissue. Once the adipose tissue is smaller, then it produces less leptin, and somehow less leptin leads to increased food intake, which then makes the clock to tick the other way around. And for a long time, it was thought that the only efferent mechanism of action of leptin was to decrease food intake. However, there were experiments suggesting the opposite, that initially, and these experiments are called pair feeding experiments. Pair feeding experiments are experiments in which we have an animal that is obese. It's obese because it's deficient of the hormone leptin. So it doesn't have leptin because of a monogenetic mutation. So the brain senses that there's no fat, and then it just triggers mechanisms to increase fat mass increase, so obesity. And these animals are hyperphagic, they eat a lot. And when we cure these animals, and also humans with the same mutation, with leptin, we see that there's a linear decrease in body weight. So these animals, they lose all the fat they have. And if this effect was solely mediated by suppression of food intake, then one would expect if we would be feeding another subset of animals that was not treated with animals with the same amount of food that leptin-treated animals would eat, then one would expect similar weight loss. But the fact of the matter is that only happens in the beginning, in the first few days. And after that, the animals that are pair-fed to leptin-treated animals but do not receive leptin treatment, they don't lose the same amount of weight. So there is a delta here. And this is not observed just once. It was observed by multiple groups in different institutes. So we thought, what is that is generating this delta? So this was the question that led me, or this was the question that inspired the beginning of my lab back in 2013 at the Gulbenkian Institute in Portugal. One glimpse into this, into the potential answer to this question came from the observation that leptin deficient patients, they have what is called orthostatic hypotension. So this is when a patient lifts up from the chair, it's going to feel a transient decrease in blood pressure. And this is thought to be regulated by sympathetic nerves that innervate the vasculature. And there's also another deficient response that these patients have, and it's called the cold pressure response test. When people put a hand in a bucket full of ice, one's going to see a deflection, a transient change in blood pressure, which is measured here in the arm cuff on the other side, on the contralateral arm. But leptin deficient patients, they have abnormal responses, although they have normal blood pressures. So this is a deficit in response to challenge, whether gravitational or temperature. And this is thought to be mediated by nerves that innervate the vasculature. And I thought that perhaps sympathetic nerves that would innervate the adipose tissue would be responsible for that delta. But the fact of the matter is, whether or not sympathetic nerves innervate the adipose tissue was a topic of contention. There was just no clear answer because there was no primary data that would be sufficiently convincing. And most of the textbooks, still to these days, they simply neglect the adipose tissue, or at least the older books that have been published prior to the last 5 years. So to address the question of whether or not the white adipose tissue is innervated, because it was known that the brown adipose tissue was well innervated, and there's primary data that is very convincing. But whether or not the adipose tissue was innervated, this was a question that we addressed not by classical histology methods that yielded data that was not very convincing, but rather by using techniques such as the clearing techniques. And in these techniques, we have a piece of fat, which is a whole adipose tissue from a mouse. In this case, this is the inguinal adipose tissue. And through a series of chemical steps that were developed by the lab of Marc Tercier-Lavigne at Rockefeller University, we were able to render this piece of tissue transparent. And if we couple this to an antibody staining that marks sympathetic neurons, and then we're able to see that the adipose tissue in a whole mount preparation that was rendered transparent, we can clearly see that there's a vast network of axons, sympathetic axons, that are labeled here against tyrosine hydroxylase, which is one of the enzymes in the biosynthetic pathway for catecholamines. And as you can see here, this is pretty convincing that these are sympathetic axons. Now, you can imagine that if you were to do an optical slice on this image, you would just have dots. And of course, those dots are not very convincing. And that's what classical histology techniques would enable you to see. Now, one could say that these nerves that are innervating a slab of adipose tissue, they're there just to subserve the biology of the vasculature. So we decided to follow these nerves, this time around with a different imaging technique. This is to photomicroscopy in an animal that expresses a fluorescent reporter in downstream of TH3. And we see here that the adipocytes that are labeled here in green, they're being subject to targeting by these sympathetic neurons. So now we can convincingly or be a bit more convinced that in fact the adipocytes, they do receive neuronal input from sympathetic nerves. Now, textbooks, they knew for a long time that adipocytes, they have the receptor that sees norepinephrine, and that norepinephrine is a neurotransmitter that sympathetic neurons release, and that we know that downstream of activation of the adrenergic receptors in adipocytes, we have lipolysis, which is the breaking down of fat. What was not known is whether the source of norepinephrine that underlies homeostatic changes in body weight and in fat mass are these neurons and not something else, for instance, the adrenal gland. The adrenal gland also produces enormous amounts of norepinephrine that enter circulation, but that only happens in a fight or flight response. So that is when you have to run away from a dangerous stimulus, or if you do surfing or parachuting or so-called adrenaline sports. It doesn't really happen if you skip a meal, or if you skip several meals, in fact. And moreover, animals that do not have adrenal glands, or humans that are injected with leptin, they can still undergo homeostatic changes in weight. So if it's not the adrenal gland, what is it? Is it these neurons? And this question bodes for a gain-of-function experiment in which we utilized optogenetics. We expressed chanoordopsin, which is a light-activated cation channel in these neurons. And we, through the implantation of an optical fiber subdermally, pointing at these neurons, we're able to activate these neurons and demonstrate that optogenetic activation locally of sympathetic neurons in fat is sufficient to drive fat mass. And through a series of experiments in which we also utilize genetic loss of function experiments as well as surgical, we demonstrated that the sympathetic neuroadipose junction mediates leptin-driven lipolysis, and that in turn they close the neuroendocrine loop of leptin action in the brain. Now, at homeostasis, the clock ticks perfectly. There's an afferent arm, there's an efferent arm, and it ticks such that it keeps weight in a very narrow range of variation. But during obesity, there's a phenomenon called leptin resistance. And in leptin resistance, the hormone, there's increased levels of the afferent arm, that is to say, of leptin. So there's hyperleptinemia. But for some reason, these high levels of leptin, they're not sensed by the brain. And therefore, there's an efferent response that it's reduced. So the idea of trying to create a mechanism or a therapy that would strengthen or amplify the efferent arm was a question that I entertained in my lab, because there was already evidence that especially from the lab of Philip Scherer at UT Southwestern, that in order to revert this equilibrium back to homeostasis, it is sufficient to tweak with one of the arms in the neuroendocrine loop. He demonstrated that by reducing leptin levels, then one can rescue normality or can mitigate obesity simply by interfering with one of these arms in the neuroendocrine loop. So Philip Scherer did the afferent arm, and I decided to do the efferent arm. And this idea would be simply that if one would be able to increase the tonic activity of these neurons or facilitate the activity of the neurons that it's lost in a leptin-resistant state, then we would be able to zap the fats in the white adipose tissue. And we could think of two alternatives. One could design a drug that directly targets these neurons solely in the adipose tissue, which is the more difficult part. Or one could perhaps target a drug to cells that would be talking to these neurons. And this is when we started studying these cells, because we believe that perhaps if these beautiful cells here that we now know are sympathetic associated macrophages, if these cells are in direct contact and if they negatively regulate the sympathetic neurons, then we would be in a good position to target these cells with immunotherapies. And to make the long story short, so that I finish up my introduction and I jump onto the single cell data. This is the role of the cells that I've just showed you. These are sympathetic associated macrophages, and they have specialized machinery to deal with norepinephrine, which is the signal that the adipocytes need to see in order to shrink and become smaller. In a situation of obesity, there's a lot more of these cells that get recruited onto nerves. And we demonstrated that abrogation of the machinery for norepinephrine uptake by these cells, abrogation of this machinery utilizing the tools of immunology, which rely on bone marrow transfers of knockouts for these transporters. So you can think about these cells as little vacuum cleaners, or in UK we call these hoovers. That have a transporter, and the transporter is the solid carrier 6A2. And by doing a bone marrow transfer of a knockout, a whole body knockout, by transferring the bone marrow, which we demonstrate to be an approach that it's sufficiently tissue specific, because no other cell in the bone marrow lineage expresses an norepinephrine transporter other than the cells that we identified. We demonstrated that this approach that spared the brain, because the bone marrow transfer cells do not repopulate the brain, but they do repopulate the immune system and lineages that derive from the bone marrow, which is the case of the cells. We demonstrated that this brain-sparing approach that is sufficient to mitigate obesity. So this gave us the idea that perhaps if we would have a drug that would function like this cork here in this cartoon, we would be in a good shape to help patients. And through a sinuous path, we ended up developing, modifying a classical norepinephrine transporter blocker, which are amphetamines. And this is the graphical abstract that, to make the long story short, we modified amphetamine by pegylation, and we demonstrated that this drug does not enter the brain to phenocopy the phenotype that we had seen in our previous approach. We demonstrated that not via the transporter, in fact, but via the beta-2 adrenergic receptor, a pegylated amphetamine drives weight loss because it promotes thermogenesis coupled to heat dissipation. And because it spares the brain, it also spares any cardiotoxicity, which is usually associated to intake of amphetamines. So this thermogenic activity is also one of the one of the energy dissipation mechanisms that is known to be part of the efferent axis in the neuroendocrine loop of leptin axons. So we demonstrated that sympathetic neurons they drive lipolysis, which makes the leptin-producing gland, that is to say the white adipose tissue, smaller. But downstream of this lipolysis, we don't have free fatty acids circulating around. We actually, the hormone in its natural state, or on the homeostatic state, it actually drives activation of the brown adipose tissue that in turn burns all the fuel that it gets released from the white adipose tissue. So white adipose. Okay, so the sympathetic neurons that are innervated, in fact, by this ganglia, or the brown adipose tissue is innervated by the superior cervical ganglia, which also have SAMs, sympathetic associated macrophages. So we asked, is it the case that there's other immune cells populating this sympathetic ganglia? And through an heroic effort by Vitaly Sarkar, who is a Novo Nordisk postdoc in my lab in Oxford, she was able to dissociate single ganglia and dissociate enough of them so that we were able to run in collaboration with Novo Nordisk single cell sequencing of these neural populations. And as you can see here, we see that there's a plethora of immune cells that live in this ganglia. And in fact, the neurons, they are actually not the majority, but I think this is a technical artifact because these neurons are very big and they're like balloons full of water. So they're very sensitive to damage, as is known in neuroscience that neurons are complicated and difficult to isolate. But see here that there's a frank population of immune cells, and we have faithful validation that we don't have cross-contamination. So we have faithful markers. We see that immune cells are only marking immune cells. They're not marking neurons. They're not marking parasites. We see that there's here faithful marking, and it's a really beautiful data set. I think that reveals high technical quality in the hands of Vitali. And we asked whether or not if this data set could give us any links or any views on what would leptin be doing to immunometabolism. As you may know, immunometabolism is a growing field, and the Keystone has had several meetings in immunometabolism. But immunometabolism studies mostly immune cells in the adipose tissue, and very rarely is mentioned that immune cells are actually creating any link with a well-known controller of adipose tissue, which is leptin. So we looked into this data set and we curiously saw that the leptin receptors seem to be expressed in this subpopulation of cells, the endothelial cells. It seems to be faithfully marking this population of cells together with other markers of endothelial cells. Of course, it doesn't mark the neurons, and the neurons are marked by faithful markers such as TH. And then when we look at the histology, we saw that these sympathetic endothelial cells, they are in fact perineural barrier cells. So we can see here it looks like a sleeve. If when you dissect sympathetic axons or nerve bundles that contain axons and also other nerves, we see that there's this sleeve-like structure. And we can look at it in different ways. So if you do a cross-section, this is how we look. They really look perineural. This is not a blood vessel. They are endothelial cells, but this is actually perineural cells that are very thin and are populating ganglia and sympathetic nerve bundles. We also did immuno-EM, and we saw that this very thin layer of cells is actually very thin, and they seem to be perineural. So we asked whether or not these endothelial cells, they would be acting like other barrier cells that exist in the gut or any other structure. So we saw that IL-33, which is well known to be a cytokine that it's produced in barrier areas in the body, it's also produced by these perineural cells. And we know that IL-33 is attractive for Tregs, and we see that the Tregs, they seem to be devoided in the brown adipose tissue of animals, conditional knockouts in the leptin receptor in these perineural cells. We see that there's a deficiency in thermogenesis, and we also see that these animals are prone to high-fat diet. So altogether, it seems that the IL-33 in specs, they confer protection for obesity independently of food intake. And this refers to the literature that already have identified neuroimmune interactions with the Tregs and macrophages in the brown adipose tissue. And we think that finally the missing piece in the immunometabolism puzzle that links leptin to immunometabolism are the specs that seem to be connecting these two different fields. So the leptin field and the immunometabolism field. The initial work was done at the Gulbenkian Institute, mostly by Inez and Roxana and Elsa. And I already mentioned the people in Oxford who took part of the spec work. And thank you so much.
Thank you very much, Anna, for this interesting talk. I have a question about the signals that cause the recruitment of the macrophages in obesity. Are these signals coming in from the adipocytes or from the neurons, or why do we have more macrophages in the obesity?
Well, we don't know yet. So this is a question that we're actively working. We still don't know what's the macrophage recruitment signal. So it's an open question. We're working on it.
Great, thank you. And then you have sort of a three-way, like a triangle of the neurons, the macrophages, and then the adipocytes, and the endothelial cells coming in the middle. So did you do any kind of formal sort of ligand receptor analysis on your single cell RNA sequencing to try to figure out who talks to who here in this?
Not yet. This is actually a very good suggestion. We haven't done that yet, but of course, I think Gitali would be interested in these sorts of analysis.
Cool.
Thank you.
Thank you very much, Anna.
Thank you.
So we're moving on now to our second talk by Nomi Khatib from the Hebrew University of Israel. The title will be Dissecting the Alzheimer's Brain from Single Cells to Cellular Community.
Thank you. Thank you for the invitation. So I will tell you today how we're dissecting the cellular landscape of the Alzheimer's brain. And more generally in my lab, we're studying the cellular and molecular changes in aging brains that are underlying age-associated neurodegenerative diseases. So as you all know, we're living in a society where the aging population keeps increasing even during these times of pandemics. And the aging-associated neurodegenerative diseases has become a huge burden in our society since we don't have any effective treatments or prevention strategies. So that's a really great motivation for us to try and understand better these diseases and try to understand them from a different perspective than what has been so far been done. Specifically, we've been focusing a lot on Alzheimer's disease, which is a progressive neurodegenerative disease. I think you all know Alzheimer's, since it's so prevalent in our society, it's actually the most common form of dementia. And at the clinical stage, there's severe memory loss and cognitive decline. And at the later stages, the damage to the brain is so severe that we have it leads to death. When we try to understand at the molecular level, we actually know a lot about this disease, but we also don't know enough. So we know that there is accumulation of abnormal protein that aggregates and accumulate in the brain over a long period of time. And these protein aggregates, so specifically amyloid beta plaques that are extracellular and neurofilament tangles that are intracellular, they are leading to damage to neuronal cells and death of neuronal cells that eventually we believe are the cause of the cognitive decline. Yet when we're looking at the last 450 clinical trials for Alzheimer's disease, we're seeing that a lot of them have been surrounding the amyloid beta plaques. We see that almost none of them have proven to be effective, which really calls for a different view of how we're studying Alzheimer's disease. And moreover, we know that Alzheimer's disease is now starting multiple years before any signs of cognitive decline. So it suggests that we should look at changes that are occurring before the severe loss of neuronal cells, other changes that occur in the brain that might be early drivers of the disease. So this is what we're doing in the lab. And we're transitioning from a more neuronal-centric view surrounding the disease pathologies to a more holistic view, looking at the entire cellular environment of the brain. So if we look under the microscope in the brain, we wouldn't see this nice, clear neuronal network, but in fact, we'll see a highly complex cellular environment where neuronal cells are surrounded by multiple other cell types that are all really critical for the function of the brain. So we have microglia cells, the resident immune cells of the brain, oligodendrocytes that are forming the myelin nerve cells, astrocytes that are important for homeostatic and metabolic balance in the brain, vascular cells, and so on. And all of this complex cellular environment is really, and the communication between the cells are really critical for the function of the brain. And we now know that there are evidence that all of these cell types are actually changing during Alzheimer's disease. And our hypothesis was that they're not only changing as a response, but rather this occurs early during disease progression, is an important driver of the cellular cascade that is leading to cognitive decline. So we want to understand what is this unique cellular environment underlying Alzheimer's disease. So this has been our goal over the past few years, and the way we've been trying to address that is by building detailed cellular maps of the brain in healthy brains and Alzheimer's brain, and in order to understand what are the cellular and molecular drivers of disease. And we've been using the single nucleus RNA sequencing technology I've developed at my time in the Broad Institute to profile every individual cell and track individual cells during this progression of the disease. So we're not the only people using single nucleus and single RNA sequencing to profile the Alzheimer's brain. And one of the stars that have emerged from these previous analyses has been microglia cells, which are been switching to a disease stage early during disease progression. We're still searching for their function in disease. What I'll show you today is how in my lab, we're actually starting from profiling tissues using all kinds of omics approaches like RNA sequencing, then building the detailed cellular maps and going back to the bench to do functional validations. But today my talk will mainly focus on our more computational aspects of our work. So I'll start by showing you what we learned so far by building cellular maps of mouse models about disease-associated glia cells in the brain, then transition to talking more about the human brain and how we're starting to identify the cellular environment of the brain by multicellular communities. So in mouse models, our work has been in collaboration of multiple labs, especially Michal Schwartz's lab from the Weizmann Institute. And we've been profiling wild-type mice in an Alzheimer mouse model called 5XFAD and profiling the hippocampus and the cortex brain regions of these by single nucleus RNA sequencing, profiling hundreds of thousands of cells. And these are the maps that we've been generating of the hippocampus, where you can see on the left the healthy mice and on the right the Alzheimer mouse model. And the different cellular populations we've captured have really profiled the diversity of cells we were expecting to see in the hippocampus brain regions, multiple subtypes of neuronal cells, and all the different types of glia and vascular cells that we were expecting to find. So overall, on a bird's eye, you couldn't see a dramatic change between healthy and Alzheimer's disease, but we could already identify the disease-associated microglia that appear in Alzheimer's that was previously found. But one thing that really kind of struck us from these more detailed, unbiased maps was a very dramatic change we could see even by eye in astrocyte cells. So we're very interested in profiling them, and we actually were focusing our analysis in later stages mainly on these cells. So what are astrocyte cells? I'll just remind you that these are cells that have multifunctional cells in the brain. I think they have the most functions out of all the other cell types. They're important for homeostasis of the brain. They're participating in neuronal function. They're maintaining the integrity of the blood-brain barrier. They're important for metabolism of neurons and other cells, and they already are known for their important role in brain repair. But for some reason, they haven't been studied that much in the context of Alzheimer's disease, even though Alzheimer's himself, Dr. Alzheimer's himself, has identified them as turning into a reactive state called gliosis at an early stage of the disease. So when we focus on our analysis on astrocyte cells alone, we could that now use diffusion map analysis to look at their transition across different cell states, since these are not discrete populations, but rather a diversity of states along a continuum. So you can see on the left the wild-type brain. We can find a range of states from a homeostatic state to a GFAP high state, which is a marker of reactive astrocytes. And what happens in the Alzheimer's brain is we see a completely new state of appears, which we call disease-associated astrocytes. And these new cells that are really highly specific to the Alzheimer's brain are seem to be not only that they appear, but we also see a decrease in the homeostatic astrocytes. So when we look at the richness of our data, we could identify that disease-associated astrocytes upregulate a number of really interesting pathways, inflammatory-related pathways, many metabolic-related pathways related to lipid and cholesterol metabolism, and also other stress response like reactive oxygen species response. We also took markers of our cells and stained the brain to showing that the disease-associated astrocytes, they're not randomly positioned, but rather strongly biased towards in proximity to Alzheimer's disease pathology. So overall, we have really strong evidence that they have an active role during disease, but we wanted to try and put this on a timeline. So we were profiling the Alzheimer's brain when there's already cognitive decline and neuronal loss. So we then repeated our analysis looking at earlier time points and later time points. And when we do that, we see a constant increase in disease-associated astrocytes throughout disease progression, and a constant increase in the homeostatic state of astrocytes. And this suggested to us that there is a transition from a homeostatic state to a disease state. This was a little surprising, since actually disease-associated astrocytes are also reactive-like. They're much more similar to the common, to the physiological reactive state we saw also in wild-type mice. This is and so we used some mathematical modeling to show that indeed in our model, we predict a transition from a homeostatic state to a disease state. So just to summarize before summarizing all our findings so far, one last important bit of information is that when we ask when do we first see the disease-associated astrocytes appear, we see them appearing early at around 4 months old mice, before any signs of cognitive decline and neuronal loss, but there is already the beginning of the Alzheimer pathologies at this mouse model. So this suggests that disease-associated astrocytes take an important role during early stages of the disease. Yet we have actually more questions than answers at this stage. So I'll just summarize what I've shown you so far. We found a very distinctive state in astrocyte cells that are not similar to the physiological reactive astrocytes that we see in normal brains. They're specific to disease. They have both linked to immune and metabolic signaling. They're positioned next to A-beta plaques. They appear early before any signs of cognitive decline, and they increase as the disease progression. There's also some kind of mild signs that they might appear also with natural aging, suggesting an interplay between the genetic basis of the disease and the pathology to natural aging. But as I said, there are more questions than answers at this stage. We're still interested in understanding the functional role of these cells during disease progression, how they interact with other cells, and of course, the relevance to human brains. So I'll just show you one more information from the mouse models before transitioning to human brains, that in our newer data, where we're starting to profile, we're going back to profiling all the different states of cells in the mouse brain and not focusing only on astrocytes, we see indeed that there is a disease-associated state across all the different glia cells. So here you can see a disease-associated state in astrocyte cells, microglia cells that I've mentioned before, and also oligodendrocytes. So this really points to the hypothesis I raised at the beginning, that the entire cellular environment of Alzheimer's is actually changing. And this is, we see in mouse model, occurring very early during disease progression. So for today, I would try to first show you that all these findings in mouse models are in fact also recapitulated in human brains, even though we're transitioning to a different genetics and a slightly different form of the disease. And so when we transition from mouse models to human brains, our challenge is actually quite big, because we have a diversity of genetics, backgrounds, lifestyles, and different environments, which all of these are actually risk factors for Alzheimer's itself. But we used a similar strategy as we used in the mouse models by building cellular profiles of the healthy brain and the Alzheimer's brain using single nuclear RNA sequencing and comparing between them. So this is, again, a collaboration with multiple other labs. I'll just mention mainly Phil de Jager and Vilas Menon from Columbia University have been working really closely with us on this. And all the results I'll show you today are from Anael Kain, my student. So as I said, we profiled 24 individuals. So these are post-mortem brains of the prefrontal cortex area of both healthy individuals and Alzheimer's individuals, both men and women, are from aging populations. And this is the map that we've been building. You can see we identify all the different cellular populations that we expect to find in the brain, multiple different types of excitatory and inhibitory neurons, all the different types of glia cells, and so on. So we can now start to actually discuss the cellular diversity that we find in the human brain, in all the different characteristics of it. And similar to what we did before, we then focused on each of these different cell types and profiled the diversity of subtypes and cell states in each of these. I'll quickly kind of summarize a lot of data about the cellular diversity to reach the more interesting part about the disease relevance. When we look across all the different cell types, we can find a diversity of states in astrocyte cells, microglia, endothelial, and multiple subtypes of neuronal cells, kind of establishing the diversity of cells in the brain. Specifically, when looking at astrocytes and microglia, we see a transition from, again, a continuous transition from a more homeostatic state to multiple different disease states, and we can link them to the same cellular markers we found also in the mouse brain. So in some sense, we're finding some similarities to the mouse brain, only we have multiple reactive states, unlike the mouse brains. And when we ask which is the most relevant state, a reactive state to the disease-associated astrocytes I just showed you, we find similarities to the fibrous-like reactive astrocytes that are also upregulating immune and metabolic and stress response signaling. So we found that very exciting. I just want to comment that not all cell types could actually be easily characterized by using discrete methods like clustering. So we also used alternative methods like topic modeling that were recently shown in order to find expression programs that are unique to that are changing across all the different cells. And every cell we now have a soft clustering approach where every cell is modeled by a combination of expression programs. And here you can see the strength of the association of each cell to each expression program. So now that we have completed our mask, we can really ask what are the disease-associated states in the human brain, and we can show some circumstantial evidence from 24 individuals showing disease association to astrocyte states or to specific programs in oligodendrocytes. But our major problem is our scalability, that within 24 individuals, we have very limited ability to connect between disease states and cellular states because of the huge diversity in the human population. So we actually used a different approach using bulk sequencing that was available for us from 640 individuals, and then using a deconvolution approach to predict in each of the 640 individuals what is the frequency of each of the cellular populations we just described using the single cell RNA sequencing. So now we have a prediction of the cellular landscape in 640 individuals, and we can now connect them to disease. So we could see, for example, cellular populations associated to a cognitive decline and to neurofilament tangles. So, for example, the oligodendrocytes programs I've just showed you, one of them strongly associates with cognitive decline, while another associates with cognitive resilience. And similar to that, you find microglia cells and others. So I'll just quickly, in the 2 minutes I have left, I'll just show you how we're now moving from single cells to entire cellular environments. So the idea is very simple, that when we look at all the cellular states that we can find in a specific cell type, when we look across 24 individuals or even 640 individuals, we see that they're not uniformly distributed, but rather specific individuals have mainly one state and not others. And you can see it here also in the 640 individual, where it's much more on a continuum of states. So we can now actually look across all the different cell types and see that in all of them, we find these interesting switches across cell states. And we can ask, are these changes actually coordinated across individuals? So we can look in combinations of different cell types and look if they're coordinated, correlated, anti-correlated, or maybe have no relation at all when we're looking across 640 individuals. So we're building these detailed pairwise correlation matrices to find these correlations across all cellular populations. And we actually really prefer to represent that using a graph where every specific node is a different population colored by the cell type, and they're either strongly correlated using the red line or anti-correlated using the dashed blue lines. So you could clearly see there is a pattern within our graph and matrices, and we can use a community detection algorithm to assign cellular populations to cellular communities. And each cellular community is more frequent in one individual and less in the others. So now we're actually divided the brain into cellular communities where different brains have more abundancy of different cellular communities. For example, we have a community of homeostatic astrocytes, microglia and endothelial cells, and others that have reactive astrocytes and reactive microglia cells. We could also try and now look at the signaling between them and find signaling that are specifically linking different cellular communities. But just to complete our story, I'll just show you how we're now linking astrocyte pathologies to our cellular communities. So we could look at the individual score for every cellular population to how much it's related to different Alzheimer pathologies or to generally cognitive decline, showing that we're finding a specific cellular population that's highly linked to all the Alzheimer pathologies and to cognitive decline. So we can now annotate our communities, showing we have a community related to amyloid beta, tau pathologies, cognitive resilience, and the homeostatic and reactive communities. So back to the cartoon representation, we're thinking that we have now the beginning of a description of how does the cellular environment of cognitive decline looks. So I'll just summarize. We started by looking at mouse brains, and defining glia-associated cells, transitioning to human brains, showing the same populations, and then to cellular communities that are linked to cognitive decline and resilience. And I'll just end by really thanking all my different collaborators and funding, and of course my lab, which is great fun to work with in COVID remote times and also when we're all together. So thank you for listening.
Thank you, Naomi. So we have a series of questions from people in the audience. So one of them is from Shauna Cooper, who asks, the penetrance of AD is nearly 100% in people with trisomy 21. Are you seeing changes within reactive cell types indicative of transcriptional changes that are chromosome specific?
Wow, that's a great question. We actually did not check the chromosomal bias, but we'll definitely do that and get back to you. It's a good suggestion.
Yeah, it seems like a cool idea. Mario Flores asks, in what cases is it better to use topic modeling instead of clustering?
So that's a good question. So we haven't invented topic modeling, but I think we're really doing a good use of it. So when you have a cellular populations that can be described by discrete clustering approaches, where you have like fuzzy borders between clusters, this would be a good place to use any approach that uses more soft clustering. And topic models is one of these approaches where you have gene programs, and every cell is modeled by a combination of programs instead of just being assigned to one cluster.
Cool. Reagan Hamill has a question, and I actually have sort of a little bit of an addendum to this. So thanks for a great talk. Have you investigated the spatial aspect of these disease-associated astrocytes, e.g. proximity to plaques, microglia, and vasculature? And actually a question that I had, I think, related to this is, so you see a change in the expression profile of particular cells, and you see some, localization difference. Now they're closer to plaques, if I got it right. I guess one of my questions is, how much of the expression profile is intrinsically different? Like the cells are intrinsically now acting differently versus, well, now I'm near something else, and in response to being near the plaque, I have a change in gene expression. So in principle, it could be all one way or all the other, and I'm wondering if you have any sense to which. But the primary question was about spatial aspect of these DAAs.
Yeah, so you actually said nicely that we did show that the disease-associated astrocytes are positioned close to A beta plaques. That's in the mouse brain. We're still exploring these aspects in the human brain using alternative methods. So we don't have, for example, the cellular communities that we found. We still need to show that they are or are not close to disease pathologies. But the question you raised about what is like an intrinsic change in cell state and what is driven by the proximity to pathologies, this is, of course, I don't have an answer for that, because what we're measuring is how it is in the tissue, and it's hard to separate between these two, but it's definitely very interesting. But I can tell you what I think. So what I think is that it really depends a lot on the cellular and microenvironment you're at, which means the secreted molecules around you. And so are these molecules coming from blood vessels and would actually affect all the tissue simultaneously? Are these really locally driven by cells that are in proximity to plaques and you're in proximity to these cells? That's something we're still investigating. And I think this is one of the more critical questions, I would say, to ask: what is the major signaling that are driving these changes?
Yeah, thank you. So apologies if I get this name poorly pronounced, but Mikhailo Batyuk asks, what is a proposed function of disease-associated astrocytes in Alzheimer's pathology?
Yeah, so we think that they actually have slightly different roles at different stages of the disease. At the initial stage, we actually think they might even be beneficial and definitely driven by the amyloid beta pathology. So that's our hypothesis. But at later stages, we think they actually are already neurotoxic on their own, and there is some evidence based on the pathways that they are expressing. And also, actually, some of these aspects are confusing because we've shown that there is a switch from the homeostatic state to the disease state. So just the lack of having enough homeostatic astrocytes to support the multiple functions that they have in supporting homeostasis and metabolism could be actually harmful for the brain on its own. So even though astrocytes might be important in responding to the pathology in the brain, just not having enough homeostatic astrocytes could be very detrimental. So we think that actually, while microglia cells are actually probably have a beneficial role at the beginning of the disease, the astrocytes will probably be the source of inflammatory neurotoxicity in later stages of the disease.
Cool. Thank you. So we will take a break now, and any unanswered questions will be available in the session forums. So, please feel free to interact with Naomi there. We'll be returning at 11:40 Eastern Daylight Time, I guess. Translate that to your time zone, and we'll see you in about 7 minutes. Okay, thank you all. Hello, everyone, and welcome back. It is my distinct pleasure to introduce my co-organizer, Shalev Itzkovitz from the Weizmann Institute, both a brilliant scientist and well in the running for one of the world's kindest and nicest people. So he'll tell us about some work on the intestinal epithelium.
Okay, thank you, Arjun. I'll tell you about spatial omics of the intestinal epithelium. In my lab, we seek to understand how cells work together within the context of tissues to collectively achieve physiological goals. And one of our favorite organs is the small intestine. It has a beautiful shape. Our gut is lined with a single layer of epithelial cells that form these highly folded structures composed of mountain-like structures called villi. They're about 0.5 millimeter in height, and deep invaginations are called crypts. And as you can see by this movie from Hans Severs, at the bottom of the crypts, we have stem cells and progenitors that continuously divide each and every day of our life, creating a mitotic pressure that constantly pushes the cells upwards. So cells here are on the move. As they exit the crypt, they differentiate into two main cell types, secretory cells or enterocytes, the nutrient-absorbing cells. And then these cells rapidly migrate up along this axis at 1 cell position per hour. And for the next 4 days, they will absorb nutrients and fight bacteria until they reach the tip, and then they will be shed off. So we shed off massive amounts of cells, about 30 grams of cells every day. It's a quarter of our feces. And what we really like about this tissue is that space equals time here. So the higher up you are along this axis, the more time has elapsed since you were born from the crypt. And that's a very nice space-time equivalence that I'll talk about in this talk. Okay, so the villus is not a uniform structure. It's a highly graded structure. There are very high levels of oxygen at the bottom, and as blood flows upwards and cells respire, oxygen concentrations go down dramatically to even hypoxic levels at the tip. On the luminal side, we have inverse gradients of nutrients and bacteria. The luminal content flows in from the tip here, and as it flows downwards through reaction to fusion convection, the levels of nutrients and bacteria decrease towards the bottom of the villus. And then we also have morphogenetic gradients, high BMP levels at the tip, high canonical wind signals at the bottom. So it's a very graded structure. And we wanted to ask whether, in response to this graded microenvironment, different enterocytes, these nutrient-absorbing cells, subspecialize in distinct functions at different villus heights. And when I say we, I mean my previous excellent postdoc, Andreas Mohr, who opened his lab in ETH Zurich lately. And so what Andreas did was to section the gut and use laser capture microdissection to catapult 5 different zones of the epithelium from the bottom of the villus to the tip into 5 different tubes. He performed bulk transcriptomics, and he found quite a high transcriptional diversity. So we found a bunch of genes that go down in the epithelium from the bottom to the tip of the villus, and some genes that go up, and we call these landmark genes. And we then use them to localize single sequence cells to infer where they were sampled from. So we take dissociated epithelium, and for every enterocyte, we examine the combined expression of these landmark genes, and we retrospectively infer what was the coordinate along the villus axis that they came from. And then we can group together these spatially stratified enterocytes, average their gene expression, and get a complete transcriptional gene expression map, which is what you see here on the left. Every row is one of the enterocyte genes, and the different columns are the different zones. So the left column is the crypt, and then we have the bottom of the villus, the middle, the tip. And you can see that transcriptionally, these cells are very, very diverse. And you could say, of course, crypt would be different than the villus because crypt contains the proliferative cells, and the villi contains the differentiated cells. But even if you erase the crypt, you can see that there is extreme diversity. And if you take a tip enterocyte and a bottom enterocyte, they're as different as an enterocyte and a secretory intestinal cell. So completely different cell states. In this visualization, you can see 3 examples of zonated genes. As the enterocytes exit the crypt over their first day of life, they express the green gene, then they shut down the green gene, upregulate the red gene, shut down the red gene, upregulate the blue gene, and then they're gone from the pit. I'll tell you just in a few slides about some of these zonated functions, because this is published work, and then I'll move to the non-published part. So as the enterocytes exit the crypt, the first thing they do, they don't engage in absorption, they actually fight bacteria. They express antimicrobial peptides that are secreted into the lumen and presumably protect the crypt from bacteria. Remember, the crypt is the holy grail of this tissue where the stem cells reside and should be viable for the organism's lifetime. There's also NLRP6 that encodes the inflammasome bacterial sensor in this zone, and a bunch of cytokines that are secreted into the basal side and specifically recruit some immune cell types into this bottom villus zone. Then as the enterocytes migrate upwards, they sequentially express the machinery for absorption of amino acids, then carbohydrates, then peptides, and at the very tip, apolipoproteins and cholesterol. And at the very tip, there's hundreds of genes that are upregulated in enterocytes just hours before they're shed off, including some key transcription factors, EGF receptor, and a very interesting extracellular protein program centered around this gene called NT5E or CD73. These are a bunch of genes that take up ATP, convert it to AMP, adenosine, and then suck up the adenosine. And all of these genes are expressed at the tip, and we think they're creating a luminal microenvironment that's depleted of ATP. Bacteria secrete a lot of ATP, and this is a danger signal for the immune system. There's a lot of immune cells here in the stroma, so there's an opening here in the tissue, and to reduce inflammation and attack by these immune cells, we think the enterocytes are reducing the ATP concentration as a form of immune modulation. Okay, so just to summarize this part, the previous picture we had of the gut consisted of stem cells, progenitors, and then what we call ternally differentiated cells. And what we think we've shown is that enterocytes are actually not ternally differentiated. They're constantly trans-differentiating. They're starting off as these sterilizing enterocytes, then switching to amino acid absorption, carbohydrate absorption, lipid absorption, at the very tip, this immune modulation. Okay, so now I'm going to switch to my second part, and I'll tell you about a question I always get whenever I give this talk, and that's the question all of us fear. What about the proteins? So I freak out when I get this question, as I'm sure many of you, because measuring proteins is very hard. But in this tissue, I want to convince you that measuring proteins is actually very important. So RNA and proteins don't always correlate because there's post-transcriptional regulation consisting of variable translation, variable protein lifetimes. But even if these regulatory rates are constant, you can have very interesting dynamics in this specific tissue. And this is because the gut, the villus, is a system that is not under steady state. If you remember, the migration time from the bottom of the villus to the top is 4 days. That's extremely fast. That's about the order of the magnitude of a typical protein lifetime, which is about 2 days. And this means you can get some very interesting out of steady state dynamics. So let's imagine our villus axis here, and this is time, but remember space equals time. And we're plotting the RNA in blue and the protein in red. If I have an unstable protein, it will closely track the levels of the RNA, right? So RNA would go up and then down, and protein will only also go up and then down. If I would sample cells along the spatial villus axis and measure both RNA and proteins, I will see a correlated behavior. So as RNA goes down, also protein goes down. On the other hand, if we have a very stable protein, we could see a very different behavior. Now RNA would decrease as the enterocytes migrate upwards, but the protein will continue to accumulate because of this for like 3 or 4 days. And if we sample single cells along our villus axis, we would actually see an anticorrelation. So these are different cells along this, in different positions. As the RNA goes down towards the tip, protein levels actually go up. So you can get very interesting dynamics that depend on the protein lifetime. So we want to measure, get the same whole genome map of both the proteins and the RNA. And Yotam Harnik, an amazing PhD student in the lab, decided to take on this challenge of obtaining these global protein measurements with high spatial resolution. And he decided to use a technique that we called spatial sorting. So the idea here is that we already have a transcriptional atlas of enterocytes along the villus axis. We're going to look for surface proteins that are zonated. And we're going to utilize them to isolate bulk populations that are spatially stratified. So I'll give you an example here. Let's say that we have the green surface marker that's only expressed in enterocytes in the crypt. and a yellow surface marker that's expressing enterocytes in increasing levels towards the tip, and then a red surface marker that's only expressing enterocytes at the tip. We can then dissociate the enterocytes, incubate them with a cocktail of fluorescently labeled antibodies against these three markers, and then we can use the green surface marker to negatively select for the crypt and retain the villus cells. And we can examine the yellow-red fluorescence, and the cells with high red, high yellow would be the ones at the tip. Cells with no red and little yellow would be the enterocytes at the bottom, and intermediate populations in these FAC spots would be the intermediate zone. This will allow us to gauge these populations, sort hundreds of thousands of cells, and then we can perform any omics measurements we would like to perform. So, for example, bulk RNA sequencing, mass spectrometry, proteomics to measure proteins, ATAC-seq, different epigenetics measurements, etc. And this is inspired by a protocol developed previously in the liver by Shani Ben Moshe and Jonathan Shafir. Okay, so Yotam converged on these three surface proteins. CD24 is only expressed in the crypt, CD26 expressed in an increasing manner from the bottom of the villus to the tip, and CD73 that's only expressed at the very tip enterocyte. And then he associated the enterocytes, incubated them with these antibodies, and this is how the FAC spot looks like. You see the correlated increase in CD73 and CD26. From each one of these gates, he sorted bulk enterocytes, measured RNA, and we compared the bulk RNA sequencing measurement with our previous single-cell spatial reconstruction. So blue is the spatial sorting, the bulk RNA sequencing, and yellow is the single-cell spatial reconstruction. You can see we have a very nice overlap. So we believe that we're sampling, indeed, these spatially stratified enterocytes. And now Yotam performed both RNA and protein measurements. And he got a very nice deep measurement of more than 3,000 proteins that allowed us to build this protein zonation map with different functions allocated in different zones. But I don't want to stall on this side because I want to immediately go to the RNA protein relations, which I think is the more interesting part of this story. So here you can see the zonation profiles of both the RNA and protein for some genes, RNA in blue, protein in red, from the bottom of the villus to the top. And you can see that there's a very nice correlation for some of these genes, like RPL27 and ribosomal protein. Ribosomal proteins go down at both the RNA and protein level. REX3B is defensin antimicrobial peptide secreted from the bottom of the villus. It's donated towards the bottom of the villus, both at the RNA and protein level. And these tip enterocyte program, immunomodulatory programs, also show correlated RNA and protein behavior. But for many, many genes, we saw an anti-correlated behavior. So you can just look at NLRP6 here, the inflammasome gene as an example, where the RNA goes down from the bottom of the villus towards the tip, whereas the protein actually goes up. So if I just look at the RNA, I would say the gene activity is higher, inflammasome activity is higher at the bottom of the villus, but the protein tells us a completely different story. And this is a very broad result. I'll just show you another visual demonstration. This is the gene STAC5A1 that encodes SGLT1, the main glucose luminal transporter. At the RNA level, you can see that it's donated towards the bottom or the middle of the villus, and it's very low RNA levels at the tip. But when you look at the proteins, you see a completely different picture where the protein levels increase towards the tip. So the zone where you have high protein expression is where you have low RNA expression, and vice versa. The zone where you have low protein expression, you have high RNA expression. About 40% of enterocyte genes are anti-correlated, are spatially anti-correlated along the villus. And this is very different than the liver, where the majority of genes show perfect spatial correlation between RNA and protein. Okay? And this goes beyond individual genes. Whole processes are donated differentially, fatty acid metabolism, peroxisomes, oxidative phosphorylation. This is just an example of respiration. If I look at the RNA for genes that encode the respiratory machinery, I would say that as the enterocyte migrates upwards, they respire less. Makes sense, they have less oxygen. But when you look at the protein, a completely different story, they actually respire more. And having this protein map was very important for us to revise the functional map along the villus axis. Okay, now I'm going to switch to mechanisms. What gives rise to this profoundly different zonation patterns of RNA and protein? And we know that RNA, denoted here by M, makes proteins through the process of translation, and we know the proteins are degraded or secreted. And both of these translation or secretion rates could depend on space. Remember, space and time here is equivalent, right? We have the differential equation. The change in protein levels at every coordinate along the villus is the translation rate times the instantaneous levels of the RNA minus the degradation rate times the instantaneous levels of the protein. So we could have a situation where these rates are zonated, and for example, at the bottom of the villus, NLRP6 is translated inefficiently, but as enterocytes move upwards, translation efficiency increases in the tip. Or conversely, it could be that protein stability is higher for NLRP6 at the tip zone compared to the bottom zone. But we could also have this discordance just because of the out of steady state dynamics. And this is what we wanted to test. So we basically addressed this very, very simple model where, remember, we measure the spatial profiles of RNA and protein. And now we can plug in different translation and degradation rates, protein translation and protein degradation rates, solve this differential equation because we have the RNA profile, and get a protein profile and compare it to our real measured protein profile. And Lisa Buchauer, a very talented postdoc in the lab, has done this with a very nice Bayesian inference model, where basically she samples different combinations of translation and protein degradation rates and calculates the posterior probability of getting the measured protein profile. Okay, so black is the reconstructed model profile, and red is the measured protein profile. And you can see that with these uniform rates, we can explain many of these discordances just simply because of variable lifetime. For example, this H2K1 has a short protein lifetime, so it tracks the protein tracks the RNA level. GSA1 has long lifetime. It acts like an integrator. RNA goes down, but protein continues to accumulate. And practically most of the genes in enterocytes can be explained by this space-independent model, which, if you think about, is very, very strange, because it means that in this tissue, RNA determines protein levels, but not in the same place and not in the same time. So a gene that's expressed at the bottom of the villus at the RNA level determines the protein levels that are needed at the villus tip, 0.5 millimeter away and 3 days away, which is a little bit strange if you think about it. Okay, so in the last part of my talk, I want to ask whether there is some kind of underlying design principle here in having so many genes where the RNA is expressed at the bottom of the villus or the middle of the villus, and the protein is expressed at the tip. And I want to go back to my first slide and remind you that this villus axis is highly zonated. At the bottom of the villus, we have high levels of oxygen. At the tip, we're in nearly hypoxic levels of oxygen. It's about 8-fold decline in oxygen concentration. When cells have a lot of oxygen, they have a lot of ATP availability. So this is the energetically rich zone, the bottom of the villus. On the other hand, the business end of this tissue is, at least in terms of absorption, is the villus tip, because this is where most of the nutrients are. And in order to efficiently absorb nutrients, the nutrients absorption machinery needs to operate very efficiently at the tip. And we know the gut is super efficient in absorbing 90-something% of the nutrients that go into it. Okay, so we have machines, the transporters that bring in the nutrients, and it takes energy to make them. Translation takes energy, and also takes energy to operate them, because you have to pump the nutrients against the concentration gradient. It just takes too much energy in a zone that is compromised for energy because there's not a lot of oxygen. So if you can split temporally or spatially split the two processes of making the machines at the bottom of the villus and then having them ready at the tip, you could optimize the function of these machines, these transporters at the tip, because you're going to have more available energy. You're not going to need to waste energy on translating the proteins de novo. Just to show you that this is the case, if we look at the protein levels of both the ribosome and the total RNA levels in single cells along the axis, which is the substrate for the ribosome, both of these go down. So the bottom of the villus is the site of massive translation. And when we look at the transporter activity or intestinal absorption protein, the protein activity, the protein amount go up towards the tip. So there's this spatial separation where protein production is at the bottom of the villus, nutrient absorption at the tip, and we believe this optimizes overall tissue function in face of these constraints of low oxygen availability and high nutrient availability at the tip. Yeah, we think we think this could explain the sort of paradoxical finding that crypt cells divide like crazy. Okay, they need to divide like crazy if we want to maintain this 4-day migration rate. And having this 4-day migration rate is the only way where you can separate these two processes. If it would have taken 20 days to migrate from the bottom of the villus to the tip, I would not be able to make proteins at the bottom and have them survive until the enterocytes reach the tip. So in order to have the separation, you need to have high migration rate that's on the order of the protein degradation rate. So I'll summarize. I've shown you spatial sorting that enables reconstruction of omics maps of the villus. I've shown you proteomics maps. We can also reconstruct the microRNA, ataxic, and other omics features. Many genes have anti-correlated spatial RNA and protein profiles in enterocytes, surprisingly. Translation of stable proteins at the villus bottom reduces load for the resource-limited upper villus zones. And we think the strategy of spatially or temporally separating protein production and protein activity could be a general design principle for other short-lived lineages, for example, neutrophils, erythrocytes, skin cells, and other tissues. So I'll end here, and I'll just thank Yotam Harnik, the PhD student who did most of the work together with Lisa Buchauer, and Andreas Moore, who did the first part, and funding sources. And we are recruiting. If you're interested in these or other projects related to tissue biology, please contact. Thank you.
Thank you very much, Aleph, for the wonderful talk. Got a bunch of questions here. So first is Reagan Hamill, who says, really interesting data. Do you think there's any way of predicting the type of correlation between mRNA and proteins from an ex vivo assay like transcriptomic data? So I guess if you lose all the spatial information, if I'm interpreting this right, is there still some way to predict the type of correlation you might see?
Okay, I'm not sure. So there are methods, independent methods that have measured protein lifetime and protein translation rates, and we did correlate. We sort of we infer these processes from the spatial separation of RNA and proteins in the tissue. There's a very nice paper from Gunnar Hansen that gavaged the amino acid, metabolically labeled amino acids, and then measured Bach proteomics and sequential days, and we have a nice correlation with that data set. There is ribosome profiling that we and others have done, and our estimates for translation rates correlate with that. So yeah, I think, I'm not sure I get the question, but there are independent methods for measuring these rates. Yeah, and many of them have their limitations.
Yeah, I think that's sort of how I interpreted that question as well. One other question that I have perhaps related to this is the inference of the prediction requires knowing like the flux along that spatial axis. So is that something that you have like a hard measurement for, or is it sort of like a fit parameter?
Yes, it's a great question. So this is sort of known, and it's about 1 cell position per hour. It's dictated by the amount of proliferation in the crypt. So the more cells divide in the crypt and the faster their cell cycle, the faster the migration rate. So basically this is mitotic pressure. And it's been measured by lineage tracing and EDU incorporation, and then measuring where the cloud of labeled cells migrate at different times. Very interestingly, I can say it's very different than human. Human, there's less dividing cells, and the villi are longer, and the timescales become interesting in human. We started to look into that.
Huh, that's cool. Okay, let me go to another question here. So, from Tejasav Sarawat at the Mayo Clinic, considering heterogeneous transcriptome protein expression in different cells in the villus, have you considered understanding the effect one group of these cells exert on another group of these cells?
Okay.
Yeah.
That's interesting. If I interpreted it correctly, so whether there's going to be protein heterogeneity within a zone, or maybe is that?
Yeah, I guess like if one cell type, I guess like how might they affect the expression in another one? So,
Okay.
and the rest of the question goes on to say, also keeping your liver zonation paper in mind, such an effect might be important for hepatocytes, which secrete many proteins. So I guess it's a question of communication.
Yes. Okay. So we really see. So first of all, we're measuring average protein expression because of the sets and limitation of the spatial sorting. Now there's the emergence of single-cell proteomics, which would be amazing when this could be applied to a tissue like the gut. I think we're going to start to see some heterogeneity within zones. The liver and the intestine, we've done spatial sorting. We've done a very similar study in the liver. Very, very different. In the liver, there's a very nice repeating anatomical unit with 12 layers, and there's no movement of cells. So we don't have this out-of-phase dynamic. And RNA largely dictates proteins. Also here, RNA largely dictates proteins, but in a very convoluted way that's transformed by these variable protein lifetimes.
Yeah. So Sveta Morali asks, well, it says beautiful story, and asks, are similar patterns of expression, i.e., from crypt to tip, conserved in vitro organoids despite the sorry, these things are jumping around. Okay. Any ideas on how mRNA protein correlations behave during development or critical periods when the GI changes? For example, transitioning from infancy to more adult phenotypes.
It's a beautiful talk, and we have a rotation student looking at that right now. So sort of the pre-weaning phase, and then the post-weaning phase. During the mouse is born with complete villi, and then you have great morphogenesis right after birth. But there's profound changes in metabolism, right? It's like a amino acid and fat diet before weaning, and then a carbohydrate diet that's more intermittent after weaning, and there's a lot of changes. So we're definitely very interested in that and looking into that, as well as high-fat diets and different calorie restriction and different diets that give rise to profound changes, even in the length of the villi. So we think these patterns could change in an interesting way.
Cool. So Francesco, perhaps I'll end with one last question. So Francesco asks, are similar patterns of expression conserved in vitro organoids despite the morphological and environmental differences?
Yes, so in mice, organoids don't make villi, and so it's impossible to test that. There's a human organoid system from Jason Spence and others that have more similar structure to the real human intestine, but we haven't looked at that. And of course, it's a great idea to look at that, and because in organoids, you can really perturb the system and really measure the effects in much more detail. So it's a great idea.
Cool. So please feel free to ask other questions, and Shalav can answer them in the chat. I'm going to introduce our next speaker, who is Humaira Hamidzada.
Thanks again for the invitation.
Thanks. We're moving now to two short talks. The first is Humaira Hamidzada from Toronto, and she will speak about single-cell transcriptome to reveal the modular macrophage structure conserved across organs and species. So welcome, Humaira.
Great, thank you for the introduction and for the opportunity to present. So today I'm really happy to share some unpublished work related to our efforts in understanding macrophage heterogeneity. So the historical perspective of a macrophage is that bone marrow monocytes enter the bloodstream, and then these blood monocytes differentiate into tissue macrophages. However, we now know that it really isn't this simple, as not all macrophages are derived from adult monocytes. In fact, there are several origins, including the embryonic yolk sac, fetal monocytes, and adult monocytes. So this presents the first layer of complexity related to macrophage heterogeneity. So the question is, where do we start our study of macrophage biology? Well, for a long time, macrophages were studied as mixed populations, but we've come a long way with single-cell technologies, which shows that even within an individual organ, there are many transcriptionally heterogeneous subpopulations that exist. However, this raises yet another layer of complexity. How are these different subsets in different organs related to each other, if at all? And this is further complicated when you consider the number of different studies with different approaches and also different species. So we do appreciate this heterogeneity, but there's a lack of a consistent approach that's limiting our ability to translate findings from one study to another. So the question that motivated our research is there an underlying macrophage organization that can inform a common starting point to study tissue macrophage heterogeneity and biology? And so we focused on commonalities between 5 organs in the mouse and human. And although there exists major tissue-specific differences, there is a very small gene profile for 3 macrophage subsets that was consistent across organs. And this transcriptional organization lent itself to track with common biological properties. So today I will be walking through the data that helped us build this common starting point in macrophage biology. So we focused on the heart, liver, lung, kidney, and brain, and we sorted total macrophages using the marker CD45, and we casted a wide net for CD64 and CD11b to ensure that we capture all macrophages and perform single-cell RNA sequencing. So starting with the single-cell analysis of mouse heart macrophages at the steady state, where we excluded monocyte and dendritic cells, we see at least 3 subpopulations of cardiac macrophages. The first subset, over here on the right in gold, expresses the genes TIMD4, LIVE1, and folate receptor 2. So for short, we term this subset TLF macrophages. The second subset in green on the other side lacked expression of subset-defining genes of TLF macrophages, but it expressed CCR2. We term this subset CCR2 macrophages. We also found a third subset in the middle here. It didn't express the subset-defining genes of the other groups. However, it highly expressed antigen presentation genes similar to CCR2 macrophages, yet it lacked CCR2. So this likely represents a more generalized population of macrophages, and for simplicity, we've termed them MHC2 macrophages. So our goal was to assess whether a similar substructure can be identified in other organs as well. So in the lung, we have this very dominant population of alveolar macrophages, but also a smaller group of interstitial macrophages, which was our interest. So zooming into these interstitial macrophages, we find at least 3 subsets that track with the reciprocal expression of TIMD4 and CCR2, and a group here in the middle that lacks both of those genes. In the brain, we have the highly tissue-specialized microglia, but also a group here termed BAMs, which stands for border-associated macrophages. And again, zooming into these BAMs, we see at least 3 subpopulations, again, tracking with reciprocal expression of TIMD4 and CCR2. This pattern is extended to the liver, where the TLF macrophages are actually the very well-studied Kupffer cells, which express the gene CLEC4F, and also extended to the kidney, where the major population are the CCR2 macrophages, and the TLF macrophages are relatively minor, yet still present. So at this very broad level, we can see that TIMD4 and CCR2 expression tracks with 3 subpopulations in 5 organs. But we wondered, beyond these couple markers, how similar are these macrophage subpopulations to each other? So to answer this question, we looked at differentially expressed genes that are found in each subset in each organ, and then we compare them across organ to assess the degree of overlap. So starting with the TLF macrophages, we see that about 75% of the subset gene signature is organ-specific. This means that those genes were expressed in one organ only, and only 11% of the genes were shared in at least 3 organs. CCR2 macrophages had a smaller percentage that is shared in 3 organs, and MHC2 macrophages appear to be the most plastic or responsive to their tissue, as only 4% of the genes were shared in 3 or more organs. Now, what's very evident here is that while we attempted to look for commonalities, the greatest signal was in fact differences, and there's only a small aspect that is similar. So we defined a core gene profile for each subset that's composed of genes that define the macrophage subset in 3 or more organs, shown here with their expression levels. So we see here that a small gene set is consistent for each subset in each organ. We then focused on the 4 top cell surface markers, TIMD4, LIVE1, folate receptor 2, and CCR2, and using random forest modeling, we showed that the expression level of just these 4 genes is sufficient to predict our macrophage classification with high accuracy in all organs. So we've really narrowed it down to just a few predictive genes. So then the next critical question was, while single cell provided us with the macrophage classification with a small conserved aspect, we wondered whether it can also track with common biological properties. So the biological work in the next 2 slides was performed by a graduate student and postdoc in the lab. So we used the top cell surface markers identified in single cell, TIMD4 and CCR2, and we assessed the macrophage subpopulations at the protein level by flow cytometry. And starting with the liver, we see that stratification using TIMD4 and CCR2 reveals 3 subsets, which expressed either TIMD4 or CCR2 or neither. And this was observed in all 5 organs, where we see that the TLF macrophages, like single cell, co-expressed LIVE1 and folate receptor 2, whereas both of the other subsets highly express antigen presentation genes and can be distinguished by the expression of CCR2. So we then use this stratification to assess if these 3 subsets differ biologically. Specifically, we assess the relationship of these subpopulations to monocytes. We use parabiosis where 2 mice are joined and they share their circulatory system. So this means that monocytes from one mouse can enter tissues in the other and differentiate into macrophages. So we first paired 2 wild-type mice together for a short-term 5-week-long time period. And after this short time period, we see that CCR2 macrophages have a very distinct life cycle, as nearly all CCR2 macrophages were donor-derived, meaning that they were rapidly replaced by monocytes. And this is very interesting because CCR2 macrophages have this very distinct life cycle, and this is consistent in every organ that we assessed. So there seems to be less replacement of MHC2 macrophages and no replacement of the TLF macrophages in this short time period. So we then assessed longer time periods, and we ramped up chimerism by pairing CCR2 deficient mice with wild-type mice and focusing on the TLF and MHC2 macrophages after 6 and 10 months. We see that there's slow replacement of both the MHC2 and TLF macrophages, and interestingly, when monocytes do replace macrophages, they do so to a finite maximum, where there's no additional replacement at 10 months in comparison to 6. We observe these very interesting patterns conserved across organs, but there are also some tissue-specific responses. For example, in the liver, the MHC2 macrophages actually do have more monocyte contribution at 10 months, and in the kidney, there is very little replacement to both subsets. So the key takeaway here is that the single cell informed a small gene profile that also tracked with conserved life cycles across organs. So I'll very quickly show some data in the human tissue that correlates with what we observe in mouse. So in human cardiac tissue in both the neonate and adolescent, at a broad level, we see at least 3 subpopulations, where the first tracks with genes in the mouse TLF macrophages, such as LIVE1. The second one tracks with the gene CCR2, and the third one expresses neither, but expresses high levels of MHC2. So this was consistent in publicly available data of the human liver, the human lung, and the human kidney. However, we wanted to assess beyond these core markers, how similar are human macrophages to those in mice. So we performed a more formal analysis using Garnet, which was published recently in the lab of Dr. Cole Trapnell. So the Garnet algorithm trains a classifier based on a cell type specific signature and then scores cells in a different data set. So we generated a signature for each subset in the mouse and scored the human macrophages, and we observed that human macrophage 1 was most similar to mouse TLF macrophages, whereas human macrophage 2 and 3 were less specific, as they were both enriched in the mouse CCR2 signature. So looking closer at the TLF macrophages versus human macrophage 1, we found that around 20 to 55% of the genes were shared, meaning that they're enriched in this subset in both the mouse and the human. So we show that TLF macrophages are the most conserved subset across species, and this is very interesting given the lack of conservation between mice and human previously reported. So to summarize, our goal was to attempt to simplify the complexity that comes with macrophage heterogeneity and to understand organizing principles that may exist. But we found that this was a very complex undertaking and that there are major differences between organs. However, there does exist a small core gene profile that tracks with 3 macrophage subsets in 5 organs. And this profile tracked with a common relationship to monocytes across organs. We hope that our work provides a starting point for those who study macrophage biology and a resource to understand macrophage heterogeneity in a range of fields. I'll end by thanking members of our lab, and in particular, the 3 highlighted here, who are the co-lead authors of this study, and our supervisor, Dr. Slava Appelman, for mentorship during this complex undertaking, and for introducing me to computational work, which has brought me here today. I'm happy to take any questions.
Thank you very much, Amira, for a really cool talk. We have time for a couple questions. So Tim Herpelink asks, thank you for a very interesting talk. Have you looked at how your three subpopulations correlate to the classical M0, M1, and M2 phenotype classification?
Yeah, so that's a very interesting question. So it has been shown that the M1, M2 phenotypes, they don't completely track with origins and life cycles, in that some of the genes that you see in the alternatively activated M2 macrophages, they do track with our TLF macrophages, so the TIMD4 live1 expressing subset. And some of the genes where the M1 macrophages are more inflammatory, where we see CCR2 macrophages are more inflammatory. However, this is very dynamic in that these macrophage subsets with different origins and life cycles, they can change in the context of disease, and they can become more inflammatory, they can become more reparative. And so we would rather propose this framework that actually tracks with biological properties, where this single cell identified a relationship, identified a gene signature that then correlates with macrophage subsets that tracks with their embryonic or adult origin and also their relationship to monocytes. And so this is a better framework to then assess macrophage function as opposed to an M1, M2.
Okay, great. Maybe let's take one more question. That was a really nice talk, Amira. There's a question about injury from Samar Tej. Have you observed if upon injury in different tissues, these macrophage modules, the CCR2 versus MHC2, replace the tissue niche and enforce differential TRM programs conserved across tissues?
So we did not assess conservation in the subsets in the setting of injury, and then all the work was performed at the steady state. However, our own lab has shown in the setting of injury the distinct roles of the embryonic-derived TLF macrophages versus the monocyte-recruited CCR2 inflammatory macrophages. For example, in the heart, we know that the embryonic-derived ones are more reparative. And so we've now extended sort of this framework to 4 other organs and proposed this as a way for other labs who study disease in other organs to assess the distinct roles of these embryonic versus monocyte-derived macrophages.
Great. Thank you very much. Unanswered questions will be available in the session forums for this talk and all of the other talks. So we're going to move now to the last talk by Michael Balzer from the Institute of Diabetes, Obesity, and Metabolism at the University of Pennsylvania. Michael, go ahead. Thank you.
Thanks, Shalev. So let me start by thanking the organizers, you guys, for choosing our abstract for a short talk and for waiving the registration fee. Always appreciate it very much. And I would like to show you some data of the kidney, where we looked at responses after acute kidney injury, highlighting cell death programs and other interesting patterns, leveraging single-cell RNA-seq. So the rationale for our study was that after an acute insult to the kidney, there is basically 2 things that can happen to your renal function. One is either it fully recovers, or 2 is there is one more or less severe way of deterioration of your renal function over time, which we call chronic kidney disease, to which the acute kidney injury then progresses. And that CKD affects 1 in 10 people worldwide, so it's a major health issue. And despite the fact that the kidney has the potential to regenerate, this potential is limited. So what we as nephrologists more oftentimes see is maladaptation and progression towards fibrosis, which is the hallmark of CKD. So the aim of our study was to identify distinct cell types, their transcriptomes and cell composition changes, on the one hand for regeneration and on the other for maladaptation. And we chose a model that allowed us to dose the degree of injury to the kidney, and we chose ischemia reperfusion injury for that purpose, because it allows us to either use short time where we clamp the the vessels to the kidney, or a long time, resulting in one, either full regeneration of function, or two, development of chronic kidney disease and fibrosis. And we analyzed 6 versus 6 mice, where we did short versus long IRI, with 3 follow-up time points, harvesting kidneys at 1, 3, and 14 days after ischemia. So leaving us with 2 versus 2 comparisons, and including also 6 control animals that did not undergo ischemia, then all analyzing with single-cell RNA-seq. So these 18 mice then left us with 160,000 kidney cells, which after quality control for excluding ambient RNA or correcting for ambient RNA, excluding doublets, and batch integrating with Liger, left us with pretty distinct clusters of all major endothelial, epithelial, and immune cells. And I'd like to highlight only 2 subsets for the purpose of this talk. The one that we always love to talk about when it comes to injury is the proximal tubular cell, of which there is 2 distinct segments, segment 1 and 3, because it's the most susceptible cell type to ischemia. And the other is the myeloid cell types, namely granulocytes, monocytes, and macrophages. Because when you look at the fraction changes between the groups, the control IRI short and IRI long, you already appreciate that these proximal tubular cells in green are decreased in numbers very, very drastically. And the second thing I would like to highlight here is the drastic increase as we go a long time after ischemia in the long, more so than in the short kidney samples, is how large the percentage gets of these myeloid cell types. So, for example, these granulocytes take up a huge chunk of the cells that we recover in these kidneys. And then looking at these most susceptible proximal tubule cells, when we subcluster those, we get a big blob of mainly healthy differentiated cell clusters with the typical markers that we would expect, and 4 other outlier clusters that are characterized by phenotypes with gene expression such as IL-1 beta, CXCL2, CCL3. So a very inflammatory phenotype, so to speak. And one thing I would like to highlight here is if you look at this orange cluster on the left, that is almost exclusively made up of cells that stem from those kidneys that were harvested 14 days after long ischemia time, so after the very severe injury, meaning that this might be really the cells that classify as maladaptive repair cells. And another way how to look at this is to visualize the cell densities that stem from these certain groups. And you see that in the control and the IRI short groups, the largest chunk of cells comes from the healthy blob, so to speak, whereas in the IRI long samples, the majority is stemming from this outlier inflammatory maladaptive cluster, which is absent in the first 2 groups, and vice versa. If you look at an example of a healthy subcluster, this is absent mostly in the very severely damaged kidneys. We then went further and did trajectory analysis, asking what kind of lineages do exist in these proximal tubular cells, employing a slingshot and diffusion maps, leaving us with 5 GMM clusters that corresponded basically to 2 lineages. And the first lineage was like this. So when we start at 1 day after injury, down at the bottom of this graph, we then have one lineage that goes along day 3, day 14, and leaves the short 14-day cells right in close proximity to the control cells, meaning they have a signature that is almost like those of the control cells, whereas cells from the very severely damaged kidneys took a different path, of maladaptation, and that was more or less recapitulated when we employed RNA velocity analysis and an IRI scoring system that we developed based on the genes differentially expressed between the different groups. We then asked ourselves, what is the gene regulatory network that drives those two lineages, inputting the genes differentially expressed between IRI short and long. And this is a scenic analysis showing the regulons that make up, that are specific for these 5 GMM clusters. And again, we had one very outlying cluster that corresponded to the one I showed you before, again, showing that the density of the regulons we found was highest in that cluster. And just to give you some examples, so one example where we'll look at the gene expression of transcription factors that try to define that lineage, that corresponds very well to the regulon activity at the end of lineage 1. And this is HNF4 alpha, TF that is very well known for PT differentiation, so healthy development and meaning regeneration in our context, whereas those other transcription factors were enriched in the second lineage or at the beginning of the damage. And if we, for example, look at this TFC-EBPB and highlight the predicted downstream targets of this transcription factor, we get again those very pro-inflammatory markers such as IL-1 beta and NLRP3 that are highly enriched in this maladaptive lineage number 2. And we then also did enrichment analysis, looking at the different KEGG pathways, for example, where we found that cell death pathways such as ferroptosis were enriched, and in the severely damaged samples, these pathways were sustained, whereas in the short ischemia samples, this was not sustained, but other programs took over, such as fatty acid oxidation or oxidative phosphorylation, meaning that is exactly what a healthy proximal tubular cell would do after it repairs from an acute injury and takes up its normal function again. So when we try to see the enrichment of some of the markers of those necrotic cell death pathways, it was interesting to find that especially markers of pyroptosis, for example, IL-1 beta, and of ferroptosis, this SLC7A11 gene that belongs to a transporter of cysteine, were highly enriched and expressed in this outlier cluster, not so much markers of apoptosis and necroptosis, for example. And lastly, I'd like to show you some cell-cell interaction data looking at ligand receptor analysis. And it was not surprising that the severity and number was increasing with the number of damage. And this is just counting, so to speak, the outgoing and the incoming connections in the proximal tubule. And lastly, I'd like to show you some of those connections when we look at the expression of ligands and receptors of these pro-inflammatory markers. We see that in the control in the IRI short, there is either not much going on, or the signaling is only within the myeloid cell compartment, whereas in the severely damaged samples, we also see expression of ligands or receptors of this pro-inflammatory signaling in PT cells. So meaning we might have a phenotypic switch of an epithelial cell now acquiring an immune cell phenotype. So to summarize, with those 2 degrees of ischemia reperfusion injury, we get 2 different trajectories. One is after short injury, we have a trajectory towards regeneration that is characterized by healthy genes that resemble control cells, that is driven by HNF4A. And when we look at long ischemia, we get a trajectory towards maladaptation that is characterized by inflammasome activation, sustained involvement of regulated cell death pathways, and an epithelial to immune phenotype switch involving IL-1 beta signaling that we're currently following up on in other studies. And I'd like to thank you for your attention and all those people involved in this project, especially my mentor, Katalin Shustak, who's spearheaded this project. And I'm very happy to take any questions.
Thank you very much, Michael. There's a sort of two methodological question. Maybe I'll group them. What did you use for the ligand receptor analysis with cell phone DB? And a question about the RNA velocity directionality, where the direction went before the branching, heading towards the origin point. So there's something confusing there.
Yeah, so for the ligand receptor analysis, we use cell phone DB, so it's a created database that most people are using. And for the RNA velocity, it's a question that we get a lot when presenting that data. I started explaining the slingshot trajectory starting at day 1 because it makes biological sense. Whereas in the RNA velocity plots, the arrows indeed were going out from the branching point in the middle. So that would be corresponding to day 3, which from a biological standpoint might also make sense because that is actually the very diverging point where the two lineages decide to diverge either into the maladaptive or the regenerative lineage. So I could try to just define the slingshot trajectory to start at day 3. I think that would not be an issue.
Thank you.
Thanks. So another question from D. Who is, what did you use for ligand receptor analysis? Did you use cell phone DB or?
Yeah.
Else? Okay.
Yeah,
so
I already yeah. Okay. It was cell phone DB.
Yeah, let me ask another one from Jonas Schulte-Schrepping. How did you subset the PT cells, and did you make sure that this very distinct PT7 cluster is not a contamination of myeloid immune cells or doublets? How many samples did you include per condition? Only one per condition and time?
Yeah, that is a very good question. So we subset all samples that we have. So that means 2 biological replicates per time point. So we have 2 animals per short versus long at each time point, and including 6 controls. So that leaves us with 18 animals in total. And in terms of the subsetting and how clean is our PT data, without going back to the first slide, it's in the general UMAP, it's a very, very clean cluster. And we were also concerned about the possibility that it might be contaminated by neutrophils that cling on to PT cells. And we excluded that possibility with employing several doublet removal strategies. And we also validated our findings in an external data set. So we find the same signature of maladaptive repair PT cells in another data set that was independently generated. So we're pretty, and it overlaps very well. And due to time constraints, I didn't have time to show this. And it integrates also very well when we integrate that with our data.
Okay, let's maybe take one more question from John Murray. Very cool talk. The ischemia-specific PT cell cluster was striking. Is this unusual, or do some or most or all other cell types also have these divergent clusters?
Yeah, it would be great to have a look at that. So far, we only looked at PT cells because it's the most important cell type if you look at ischemia. But for sure, I would expect that if you looked at the loop of Henle or the distal tubule, that we would get probably similar responses in terms of the trajectories. But that is that we haven't done yet.
Okay, thank you very much, Michael.
Thank you.
And I'd like to thank all of the speakers for this session. We encourage you to continue the conversation on the dedicated session forum for this topic, where you can continue with Q&A with the speakers and share insights with fellow audience members throughout the course of the meeting. Also, tune in to the joint career roundtable discussion in about an hour, I think at 2:30 p.m. Eastern Time, for career development insights from field experts. And check out the interactive e-posters today at 3:30 p.m. Eastern Time. where you can talk with the presenters, ask authors about their work live on the Remo video platform. So thank you very much.
Yeah.